One important role that eukaryotic gene regulation plays in eukaryotes, especially multicellular organisms, is in determining different types of cells or determining cell types. And one common theme, one theme that's pervasive all the way from yeast to humans, is that the choice of cell type or deciding which genes to turn on in which cells often happens in a combinatorial manner. And one really good example of how this happens is in the choice of yeast mating types. Now, yeast can either exist as haploid cells or as diploid cells. Haploid cells can come in two types, two mating types. One is called alpha and the other is called A. Now, the in the haploid state, the yeast can undergo mitosis and multiply, and the alpha colonies will multiply and make more alpha cells, whereas the A colonies will multiply and make more A cells. However, it's also possible for the alpha and A cells to fuse, so in effect carrying out fertilization, if you will, to create a diploid A over alpha cell. And this diploid cell can also undergo mitosis, and you can have a colony of diploid yeast cells, but they can undergo meiosis and give rise to haploid alpha and haploid A cells once more. Now, it's clear from looking at this that these different forms of yeast must be expressing different combinations of genes. For example, some genes must be common genes, which all three types express. For example, alpha and A and diploid all undergo mitosis, and therefore the genes involved in the cell cycle and mitosis must be common, must be getting expressed in all three types. On the other hand, the genes having to do with fusion, for example, would be haploid specific, since only haploid cells can undergo fusion. Furthermore, there must be alpha specific genes as well as A specific genes that make alpha and A distinct from each other. And so one could try and see which of these classes of genes would be expressed in which cells. So let's say I have an A cell, and this cell should be expressing A specific genes, it should be expressing haploid specific genes. However, it should not be expressing alpha specific genes. Similarly, if I have an alpha cell This alpha cell should be expressing alpha specific genes, haploid specific genes, but not A specific genes. And finally, if I have a diploid cell, so an A over alpha cell, this cell should not express haploid specific genes since this is a diploid cell, nor should it express A specific genes or alpha specific genes. All these three categories of genes should be off, and the only category of genes that will be on are the common genes. So, although it makes sense that we should have this general scheme of gene regulation, how is this actually achieved? It turns out that there's a location in the yeast genome called the MAT locus. Now, in an A cell, the MAT locus carries a gene called A1, whereas in an alpha cell, the MAT locus has the genotype that carries two genes, alpha 2 and alpha 1. And of course, if you have a diploid cell, then you will have two copies of the MAT locus, and you will have A1, alpha 2, and alpha 1. Now, there is one common protein called MCM1. So MCM1 is one of those common sets of genes that we discussed that are expressed whether you are diploid or haploid, A type or alpha type. And together, these different combinations of proteins that are available in each type. So MCM1 and A1 in A cells, MCM1, alpha 2 and alpha 1 in alpha cells, and MCM1, A1 and alpha 2 in diploid cells control the transcription of thousands of genes across the yeast genome, so that the A specific genes are on only in A cells, alpha specific genes are on only in alpha cells, and all A specific, alpha specific, and haploid specific genes are off in diploid cells. In A cells, MCM1 binds to binding sites upstream of A specific genes and turns A specific genes on. The alpha specific genes are off since there's no activator activating them, whereas haploid specific genes are on by default. When you are an alpha cell, then the A1 protein is not expressed, and instead you express alpha 2 and alpha 1 proteins. The alpha 1 protein binds to MCM1 and recruits it to the upstream activating sequences of alpha specific genes or the enhancers of alpha specific genes, and turns alpha specific genes on in alpha cells. Whereas alpha 2 works just like GAL80 works to prevent MCM1 from activating the A specific genes. And therefore, even though MCM1 can bind the enhancers of A specific genes, it's prevented from activating them by alpha 2. And therefore, in alpha cells, alpha specific genes are on, A specific genes are off, and haploid specific genes, which are on by default, are also on. Finally, we come to the diploid cell type. Now, in a diploid cell type, in principle, we can express three proteins, A1, alpha 2, and alpha 1. However, the alpha 2 and A1 proteins, they work as repressors and they prevent the transcription of the alpha 1 gene. So even though the diploid cell has the coding sequences or the DNA that encodes A1, alpha 2, and alpha 1, only A1 and alpha 2 are expressed, since A1 and alpha 2, they bind to each other and work as a repressor to shut the transcription of alpha 1 down. Now, in the absence of alpha 1, MCM1 is not recruited to the enhancers of alpha specific genes, so alpha specific genes are off. Alpha 2 is present in the cell, and therefore alpha 2 works to prevent the activation of A specific genes by MCM1, and therefore A specific genes are also off. And all the haploid specific genes have DNA sequences that bind the heterodimer A1 and alpha 2 bound together, and these two proteins work as repressors, just like when they shut alpha 1 down, they shut all the the haploid specific genes down. And so yeast choose what type of cell they are, whether haploid or diploid, A or alpha types, by expressing different proteins depending on the genotype of the cell, and then turning genes on or off using different combinations of protein complexes, different combinations of proteins that bind the enhancers or the DNA sequence that regulate transcription. And this combinatorial control of the choice of cell type is very common and occurs in multicellular organisms as well.